Hello, students. We are in Module 3 of our online hybrid anatomy course, Exploration Adventure, and today we are going to talk about cell organelles and cell differentiation and just a teensy bit about cancer and how it comes about. So we actually aren't going to talk about crossing the membrane. I meant to take that out. That's going to be a whole separate module because it's a little complicated. But we are going to talk about the following parts of the cell: the nucleus, blah, 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 blah. We'll get through every one of these. We'll talk about why they're relevant to different parts of anatomy. We'll talk about how cells differentiate. So if they all have the exact same DNA, why are they different? Why is a skin cell different from a bone cell? Why do we not grow stomach cells on our face? That kind of thing. And then finally we'll end with how cancer starts sometimes, and the difference between benign and malignant tumors. And yeah, let's just get started. So let's first talk about, here we go. Let's look at a cell in general. So if you just kind of look at the general cell model, it's a very generic version of an animal cell. And since this is human anatomy, that's all we care about. It's a human cell or the animal cell. So not all cells look exactly like this, but it is an easy way to kind of think of a general cell. We're going to look at neurons and muscle cells and how they're different once we get to their specific units. But in general, we can kind of oversimplify how a cell looks by looking at it in this way. And all cells are surrounded by a cell membrane. And so this cell membrane is made of lipids. And we talked about lipids in the last module. A lipid is a hydrophobic molecule. It is going to assemble, a lipid in general is a hydrophobic molecule. The phospholipid layer, the plasma membrane, is made of two layers of phospholipids so that the hydrophobic ends face the environment, so either the internal environment of the cell or the external environment of the cell, but the hydrophobic ends sit on the inside so that they are completely away from water. And these things self-assemble, and so they're not held in place by anything except for the fact that the hydrophobic ends want to be on the inside away from water, and the hydrophilic ends, the ones that are polar, are on the outside where they can interact with water and other polar substances. So that's what differentiates the cell from the rest of the environment. So the cell is surrounded by this phospholipid bilayer, or the cell membrane, or the plasma membrane. Those things are all the same. And on the inside, at first everyone thinks, oh, it's just a big bag full of goo, and that goo is called cytoplasm, and that's the watery environment inside the cell. And for a long time, that's all we thought they were just big empty cell. Like, they're called cells because the guy who discovered them, Robert Hooke, looked at them and they was like, hey, it kind of looks like a honeycomb or a monk cell. There's nothing in there, because when he created cork cells, that's what he thought. He thought he couldn't see well enough in his microscope to see what other stuff was going on in the cell. But we now know cells are incredibly complicated, but they are, the fluid that fills a cell is called cytoplasm. Okay, so this means cell goo, basically. And so the cytoplasm is the fluid that fills the cell. If anything's floating in the cytoplasm, we're talking about it just floating in the watery environment within the cell. Anything outside the cell, we like to talk about the extracellular matrix. So if it's extracellular, it's outside the cell. And so usually there's a scaffolding of some kind of protein system that kind of helps the cell stay in its place or move around or help stuff get to the cell, and that's called the extracellular matrix. So if it says extra, we're talking about outside. If we're talking about cytoplasm, that's the fluid inside. We are going to talk a bit, and we're going to see a little video at the end that's beyond the scope of this course, but it's interesting to watch at, that talks about a lot of stuff going on in the cell. But the plasma membrane also has some cholesterol in it, which is why you have to actually take in some cholesterol in your diet, even though for a long time they told us cholesterol is bad, don't eat any, you'll die. Wrong. And we actually need a little bit of cholesterol at least. There's also there are protein channels that actually span the plasma membrane, so that stuff can get through the plasma membrane, even if it is the inner part of the cell of the membrane is hydrophobic, but you can pass through the membrane using these gated channels or these, we'll talk about that later. We're going to get back to that when we talk about osmosis, diffusion, and cell membrane transport. But you can see stuff can kind of sit in the cell layer and do something, and that's really important. And these are usually proteins We talked about how proteins do a lot of functions. These might be enzymes causing something to happen. These might be just channels, or they might be structural. They might be helping cells stick together. There's a lot of different things that could be happening there. Really, what you need to get from this slide is that the cell is surrounded by a plasma membrane that separates the internal environment from the external environment. And in order to get through that layer, a lot of times you have to have these proteins that sit in the layer and help stuff get through. And we'll talk about that later. All right, so let's go inside the cell. So the very first thing you'll see inside a cell, if you go back here, is this giant center here. It might actually not be. You might see some other stuff, but we like to jump straight to this center part, which is called the nucleus. We talked about a nucleus. We talked about atoms. The nucleus of the atom contains the protons and the neutrons. The nucleus of the cell contains the DNA. And so, if you get into it, there are many nuclei, which is the plural nucleus, in the brain. For centers of a certain something function. Nucleus just means center. So in the nucleus of the cell contains the DNA. So let's look at that a little closer. You can get, so if we look at the cell membrane we just talked about, and we get a little closer, the center, the nucleus also has its own membrane. We just call it the nuclear membrane. Makes sense. We can use certain stains when we're looking at cells to just stain the nucleus so we can see it really well. And what's happening inside the nucleus is this is where the DNA is all the time. In animal cells and in human cells, the DNA, the nuclear DNA never leaves the DNA. Leaves the nucleus. This is like having the perfect, like your original copy of a book, and you want to make sure that this book is protected forever. You're never going to just give your book away. You might make a copy of a page, and then someone can take that copy away, but you're never going to give that book away. So the DNA inside of our cell is like the precious. You don't want to mess that up. You don't want that just being accessed by anybody, any proteins, any lipids, whatever. So you keep it inside the nucleus and very tightly regulate what can access it. And so that's very important, that all the DNA is stored in the nucleus. The nucleic acids are made here, so the DNA and the RNA are made here, but the RNA can exit through nuclear pores. And depending on what kind of RNA we're talking about, it has different functions. The entry and exit into and out of the nucleus is not random. It depends on, different kinds of RNA will enter when it's time for a cell to do a certain thing. Different kinds of RNA will leave when they are ready to go get processed to do something else, that kind of thing. So it's not just random stuff entering and accessing the DNA and doing whatever it wants. It has to be very tightly regulated, because the DNA is where your genes are, and that's what determines what your cell is going to do and who you are and what's happening to your body and all these different things. So you definitely want to make sure that's not just randomly, growing ovaries on your face. That would be a bad thing. So your body tries really carefully, your cell tries really carefully to really regulate what is happening in the nucleus at any one time. So there's this thing called the central dogma of molecular biology, the central dogma of biology, and that is that the DNA, so this determines everything about your cell, your body, your heredity. DNA, what it does is it'll make a copy of itself, so that's DNA replication. Okay, so because when cells are dividing, you want to make sure you have a perfect copy of that DNA to go on to the next cell. And then so that's just called replication. And then whenever the DNA is, it needs a working copy, that it makes RNA, and that's called transcription. So DNA makes copy itself, called replication. DNA makes little versions of itself, like little, if DNA is a book, then RNA are pages out of that book that go do stuff. So if you have a piece of the DNA that you want to use, you want to express a gene, what has to happen is it's not just good enough to have the gene for it. You have to then have, that gene has to be activated, basically, by saying, I'm going to make a little copy of it that can then leave the nucleus, so that's the RNA. And remember, RNA is a single-celled nucleic acid. It's slightly different from DNA in that it uses a uracil instead of a thymine base. Not super important that you know that. But what it does, it goes and copies just a little bit of the DNA that you want to use. It then leaves the nuclear membrane, and it goes onto the cell itself, the rest of the cell. the cytoplasm, and it can then be translated. It goes through translation, and when it does that, it goes from being a nucleic acid into a protein. So it's like it's being translated into a new language, and it's that protein that actually does whatever job it is that that gene is coding for. So RNA is basically a temporary blueprint that's used to carry information from DNA to instruct ribosomes how to build a protein. So ribosomes, we're not really going to talk about them a lot, but basically they are the functional, they're kind of an intermediary that makes sure whatever this RNA says gets turned into a protein. So the ribosomes are the ones that bring in the amino acids and make sure that they get ordered in the correct way. All right, so that is the DNA. So if you then have a little bit, well, okay, let's first go on. The endoplasmic reticulum, these are membranes in the cytoplasm, which form a separate component or compartment within the cell. So they're basically an infolding of a bunch of membranes. And what they do is you have two different flavors. You have rough endoplasmic reticulum, which is abbreviated RER, which is almost as hard to say as rough endoplasmic, oh my goodness, see? Rough endoplasmic reticulum. Sometimes we just call it the rough ER, it just makes more sense. It makes it easier to say. And then there's a smooth ER. The only difference is that the rough ER has ribosomes attached to it, which are these little combinations of protein and RNA, which are used, a different kind of RNA, but they are used to bring in amino acids and interact with the messenger RNA that's coming from the DNA and actually make the protein. So they're sitting here waiting for the mRNA to come, or the messenger RNA to come by and say, I've got a gene, let's go, let's make this happen. And then those ribosomes make peptide chains, basically translate peptide chains from that RNA sequence, and they transport them. So the rough ER, or the endoplasmic reticulum, is then going to say, oh my god, now we have to make sure that these these peptide chains get moved on so they can be processed further. They're not just a bunch of beads on a string, or they are actually, which is also DNA reference, so don't worry about it. They're not just a bunch of amino acids in a string. We need to be able to fold them into certain ways so that they can be processed further. So the first step is they go through the ribosomes, and then the ribosomes with that attached, come over to the endoplasmic reticulum, and then the endoplasmic reticulum figures out what to do next. The rough ER. The smooth ER doesn't have any ribosomes, and what it does instead is basically the same idea as where the rough ER is figuring out what to do next with the peptides, those peptide chains. The smooth ER is figuring out what to do with nonpolar molecules like steroids and lipids. So your mitochondria, which we'll talk about in a second, makes the steroids, and then they may go to the smooth ER to figure out where else they're going to go. So they might get tagged with certain things that tell the body, this can only go here. There may be other lipids also that say, oh my god, you better send this to the liver right now. And so that, if it doesn't go through the smooth ER, they could just end up in a random place, and then the place in the body that needs them won't get them. Alright, so here's what they look like if you were to slice a cell and do what's called transmission electronic microscopy. And so if you look straight across, you see all this folding. Oh my god, you see it actually takes up a huge portion of the cell. And that's just in this specific kind of cell, but it's really important that if this is your nucleus and it has a ton of DNA in it, and that DNA codes for a protein that's actually going to make something happen, you want to make sure that protein is folded and sent off properly, or else weird stuff might happen. And you see that in disease states a lot. So this is what a ribosome technically looks like. It's got two parts to it. So if this is a piece of mRNA called the messenger mRNA that's copied off the DNA, so if we were to look back over here, the mRNA, so this is actually getting ahead of ourselves a little bit. So if you have a DNA molecule, you have DNA in the center of your nucleus, or in the nucleus, then you say, oh no, I need this gene in order to make a certain, let's say to make brown eyes. I need to make sure I'm making the protein that makes brown eyes. So the mRNA gets copied off the DNA, it gets transcribed off the DNA, and says, I'm the brown eye protein, or whatever this gene needs. It then leaves through these nuclear pores in the membrane, and as soon as it leaves, it gets attached to ribosomes, okay? So that's what's happening here. So ribosomes, there's two parts of the ribosomes. They basically clamp down on the start of mRNA like a sandwich, and then they read across, and it's basically like zipping on a zipper. Okay, it's the zipper part, and this is like this is like the pull on a zipper, and this is the zipper part. Except the difference here is instead of zipping two parts together, when the ribosome zips or travels along the mRNA, it starts bringing in amino acids. So it reads sections of 3, and it says, oh, these 3 codons, or these 3 nucleic acids, specifically code for one kind of amino acid. So I better go get that amino acid. And then this one codes for a different amino acid. And then this one, It goes on, and then you see as this comes out, you get a chain of those amino acids which are stuck together with peptide bonds. Okay, and so as this is reading the mRNA, so you can see if this is the purple, the dark green, a lighter green, a lighter green. Oh, look, here's the one that goes to it. Here is a green that goes with that one. Here's a light green. So this codes specifically for that amino acid. It says, oh my God, go get this amino acid. Put it in this order. Oh, goodness. And then the ribosome. So the ribosome is basically the, since this is called translation, it is the translator to make sure whatever language this is, so the nucleotide language, gets translated into a protein language, which is what actually does the action in the body. So in the ribosomes, proteins are made in the ribosomes. Ribosomes themselves are made of 2 small subunits, which are a mix of ribosomal RNA and some proteins, some ribosomal proteins. So there have to be ribosomes to build the proteins for ribosomes. Kind of a brain explosion. So a lot of times ribosomes are just kind of existing. They're hanging out in the cytoplasm, or they might be attached to the endoplasmic reticulum in that rough ER. Let's see what else. Ribosomes, DNA, and RNA are made in the nucleus. Okay, all these things, because these are basically nucleic acid, so DNA is nucleic acid, RNA is nucleic acid, ribosomes contain a nucleic acid. So in order to have a functional ribosome, you have to make the RNA part of it in the nucleus, but then the protein part of it is made in the cytoplasm. And so these are totally made in the nucleus. Part of this is made in the nucleus, and then the protein that goes with it is made in the cytoplasm. So all proteins are made in the cytoplasm by ribosomes. Hint, good thing to know. Okay, and again, this is our example. You have RNA, it comes out. As soon as RNA comes out of the nucleus, it gets clamped onto by a ribosome, and you could actually have another ribosome sitting here and another ribosome sitting here. So you're making a whole bunch of the same protein off of one messenger RNA so that you get a whole lot of signal out of one message. Okay, what's this? Ribosome. Okay, so the ribosome, if it's docked to the rough endoplasmic reticulum, it might feed, and this happens sometimes, it feeds that polypeptide chain into the endoplasmic reticulum, the rough endoplasmic reticulum, and then it starts to gather all of the same types of proteins, or the same polypeptide chains. It bends it into the specific orientation it needs to be functional, and then it keeps it in the shape by creating disulfide bonds or cysteine bridges, basically chemical reactions between different parts of it to fold it into the certain shape. And then once it has a whole bunch of them, it buds off a little transport vesicle. And we're going to see a video where you see a transport vesicle. These vesicles, or these little buds of cellular membrane space, can be full of certain kind of proteins and then have like basically signals or tags on them that tell the cell, hey, make sure this endoplasmic reticulum gets dumped outside the cell because maybe it's a neuron and these are neurotransmitters, or make sure this ends up in the bloodstream because it's a hormone of some kind. Whatever it is, you basically are going to take, as soon as, if you have a whole bunch of ribosomes feeding into this as an endoplasmic reticulum, the polypeptide chain, or this pre-protein, will have the very ends of it will say what it needs to be done with it. And if it says go to the rough ER and put it into an into a vesicle, or this little bubble, it'll do that. So the rough ER makes sure that those protein parts get put into whatever specific vesicle they need to be in. So basically it says, do I need to put it in a certain envelope and send it somewhere? Well, then I better do that. So another part of the cell that does this is called the Golgi apparatus. It's this curvy, flat, kind of squishy, pancake-looking thing. It also has vesicles that pop off of it, just like the endoplasmic reticulum. it takes much more mature proteins, and then it says, oh, so now we have this very complex protein, this very complex enzyme or something, and we should dump it into the bloodstream, or we should make sure it gets to another part of the cell, or whatever. So maybe you just made a whole bunch of insulin proteins, and you need to make sure that they're saved for when you need them. So you might keep them in a vesicle. So the Golgi apparatus will sort these and say, here's a whole bunch of insulin molecules. We're just going to sit on them for a while until we need them. So this vesicle might originally come from the endoplasmic reticulum and say, oh my God, we have some pre-proteins that look a lot like insulin. And then the Golgi says, well, let's do some fine tuning to make sure they're really insulin, and then we'll store them in vesicles until we actually need them. So you might have heard the Golgi apparatus is like the post office of the cell, because it'll put on its little vesicles that are popping off full of specific things. It'll put little tags that say this needs to go here, this needs to go here, or save it until this signal happens. So it's postmarking, and also you could have a vesicle that's full of something that says we need to kill this certain protein on a certain deadline, or else it could go bad. And so it's postmarked. It's also marked for where it needs to go in the body because of what the Golgi apparatus does. And so this is what you might see. You have, so here's the nucleus. The nucleus is full of a bunch of DNA. The DNA then lets out messenger RNA, which goes out into the cytoplasm. The ribosomes that sit on the rough endoplasmic reticulum grab that mRNA and start making peptide chains translated off of it. So those peptide chains, so if we go down over here, they could be put into vesicles. They pop off little bubbles or vesicles, and they say, I'm full of protein, oh goodness. And then that vesicle goes to the Golgi apparatus and says, all right, I either need to hang on to this protein for a certain amount of time, or I need to finalize any kind of final production of that protein within from what it already was, and then I'm going to send it out of the cell in what is called exocytosis, where that vesicle then binds with the cell membrane and then basically throws all of its proteins out into the extracellular matrix. Usually this is into the bloodstream. If let's say this is inside the stomach or an intestinal cell, it'll release digestive things into the inner part of the organ or whatever it is. So you started with a nucleic acid here that was saying, here's a sequence of nucleic acids that makes me a person. And then, but in order to read those nucleic acids, which are only 4 different nucleotides, you create a messenger, which then goes into the, which is the messenger RNA. The messenger RNA is read through a ribosome, which is feeding that amino acid chain into the endoplasmic reticulum. And so it refines it a little bit and sends it off in a vesicle that goes to the Golgi, which finishes refining it and then sends it off where it needs to be. All right, so what's a vesicle? There are different kinds of vesicles, those little bubbles that pop off. These are basically borrowed phospholipid bilayer membrane. So you have this big membrane, you just pop off a little bit, bloop, and then you keep it. It could be, there's different kinds, okay? And we're going to look at a video that shows how these vesicles are moved. They don't just kind of float off like soap bubbles that you might make in the air. They follow very specific tracks, but those are hard to draw, so you usually see them in a book just written as little bubbles that float around. They don't actually do that, but what might happen is you have a cell that's taking in a food molecule, and it's got a tag on it as it takes in that food molecule that says, this is food, oh my god, this is food. And you have what's called a lysosome. A lysosome. A lysosome is a vesicle that specifically contains digestive enzymes. And so what it does is it merges itself like 2 bubbles coming together with that food vesicle. It digests the food into what it can actually use, and then it can let the cell use the rest of that stuff. So a lysosome is a small sack of enzymes, which remember is a kind of protein, which break down lipids. Carbohydrates is what this says. We should make that a 2, so we know it's twice as many hydrogens as oxygens, and proteins. So lysosomes break down lipids, carbohydrates, and proteins. Probably also nucleic acids, but they don't say it here. It is basically the digestive system of the cell. It's also called a suicide packet because it can also go and it kills certain things. So if you're a protein that gets put into a lysosome, you are basically, the cell is done with you and it's getting rid of you by putting it into a lysosome. All right, so that's we've already started going through a lot of things in the cell. We talked about the nucleus, the endoplasmic reticulum, the ribosome, some different kinds of RNA. We talked about the Golgi apparatus, but let's talk about the mitochondria. So the first thing you learn in like 6th grade biology is that the mitochondria is the powerhouse of the cell. But what does it mean to be the powerhouse of the cell? So within the cell, you have these oval-shaped guys. They're almost oval-shaped organelles, so teeny tiny little organs inside the cell. They're interesting because they have their very own DNA, and you only get it from your mother. That's because the sperm cell has to be so small that the part of the sperm cell that actually enters the egg doesn't contain anything except for the nucleus of that sperm cell. So no endoplasmic reticulum, no Golgi, and no mitochondria from the sperm cell enters the egg. So your egg has mitochondria, so you only get your mitochondria from your mother. And there's DNA in there. It's separate from the nucleic DNA completely. And you can do interesting mitochondrial studies to follow maternal lines through history, which is interesting. We can talk about the ramifications of that for animal population studies if you want to. Just let me know. They're very interesting. But what you really need to know about the mitochondria is that this is where energy production happens. And we're going to talk about this more specifically when we talk about cellular respiration. But this is where ATP is produced in mass quantities, and ATP is the energy molecule of the cell. It is the energy currency. It makes stuff happen, and it is created in the mitochondria, which is why it's so important. So the mitochondria has an outer membrane and then an inner membrane. And it's very important because that's where a lot of cholesterol is turned into steroids. It's very important. You also see well, we're not really going to talk about it, but there's something called the electron transport chain, which happens through the membranes, and that is why that's extra important. There is a theory out, remember in sciences, a theory is something that's well supported, and everybody knows it to be true. There is, I don't know if it's a theory or hypothesis, we're still gathering evidence, I guess, as a scientific community, but that what happened was you had two, way back long before humans were a glimmer in the future, all you had in the ocean were a bunch of single-celled organisms. They were eating they were either algae type things or algae that were taking in sunlight and using that to create sugars and then feed themselves, or they were animal cells, which means all they were doing is going around and eating the plant cells. Well, what is thought to have happened is some animal cell ate this cell, and instead of digesting it and then like it normally does, it basically co-opted or stole the reproductive power of that cell, because that cell was giving off so much energy that whatever cell had originally eaten it was like, oh my god, if I just it's basically like farming. And so it took the mitochondria, whatever the original mitochondrial organism was, and said, instead of just eating that, I'm going to keep it and feed it and give it a safe place to live so that it'll just keep giving me all this energy. So it's basically like if you had a goat and you could either kill that goat or you could keep the goat alive and use its milk, what's going to give you more food for a longer amount of time? That's what's happening here. The larger cell said, I'm going to keep this mitochondria and I'm going to make sure it's well fed and safe so that it gives me lots of energy. And that is an interesting theory, and it totally makes sense because the DNA is so different than our nuclear DNA. Okay, so like I said, the inside of a cell is not just empty space. It's not just cytoplasm. There is cytoplasm that fills the cell. There's actually all these tracks that stuff rides on inside of the cytoskeleton. We're going to see a video on this at the end. It's basically these long proteins, including actin, some intermediate filaments, and some microtubules that allow for stuff to basically zoom along like on a train. So if you make something in the nucleus and you really need it to get to the outside of the cell, if you just have a lysosome, or sorry, not a lysosome, a vesicle just pop off of the Golgi apparatus or something, the likelihood that it'll just randomly float and get to the outside of the cell, it'll happen, but it'll take a long time. Whereas if you throw it on a tract and then push it in that direction, it'll happen a lot faster. Yep, okay. And there are also parts of this cytoskeleton can then project out of the cell into things such as flagella and cilia. So cilia is basically like this. I was thinking of it as like a fur. It's like if fur could do things like a bunch of little fingers that stick outside of the cell. So they're projections from the cell membrane. The cilia are small. There's a whole lot of them, and they line stuff like the respiratory tract and the fallopian tubes. So your respiratory tract, your trachea, and your bronchi that go into your lungs, they have all these cilia lining those tubes. And so if you cough, or what you're doing is you're clearing the mucus out of your lungs, but the mucus is always moving through your lungs because the cilia are basically pulling it up strand by strand. So the strands are moving, pushing that mucus up and up and up until it gets to the top of the trachea and shoves the rest of that mucus into the esophagus to go into your stomach and be digested. If you're a smoker, nicotine kills these and they just die, and all the cilia die. So sometimes a lot of smokers cough is just because you don't have any cilia helping move that mucus layer out of your lungs. That mucus layer is really important because it traps dust and other things. But then it gets dirty and you have to get rid of it. But if you are taking nicotine of any kind, the cilia die. But if you stop taking nicotine, they start growing back within a month. My dad was a smoker from when he was 15 until like last year, he was 59. And after like a month, not just a month, he's like, yeah, I can really breathe. I can really feel. I don't cough as much. It's really weird. Like, yes, that's because your cilia has come back to life and you aren't worried. You don't have that horrible cough anymore. But he said it was meditation. Whatever. It's really actually the cilia coming back to life and moving the mucus, but whatever it takes to get people to stop smoking. The only place in the human body that we have flagella is in the sperm cells, and all the function of the sperm cells is that they're very long, or sorry, not the function of sperm cells, but the function of the flagella, or the tail on the sperm cells, is they turn really fast and it moves the cell through the cytoplasm. It moves it through the extracellular matrix or through the uterus very quickly, which is important. We'll get to it when we get to reproduction. We'll also get to why the fallopian tubes are full of cilia as well. But let's talk about, speaking a little bit of reproduction, how we go from one cell to many cells, because we very clearly, as organisms, have many cells. So what the heck is the point? Why do we even care about cell division? Well, first of all, it's important to know about cell division because cancer has become such an important issue in medical life now. It's also very important to realize that if you're going through reproduction, you are producing cells that are then being differentiated to create a new person. And you also do this as you're growing and developing. You do cell production every day of your life. So just because you've stopped growing and your growth spurt is over, doesn't mean your cells are not continuing to reproduce. And so that's another really important reason to understand what's happening here. Because anytime a mistake happens, something bad could happen. And so this is a good place to make sure you're checking for pathologies. So there's different things going on in cell division. So cell division itself is the making of more cells. Apoptosis is when you remove cells. That's not necessarily a bad thing. So apoptosis is called programmed cell death as well. But if you think about if you have a fetus and it's growing hands, you need to, first of all, the hands are kind of webbed, and then or the little pre-hands basically are webbed. And look at this giant bulge here on the forehead. That's also not a final person. So the parts of the body that don't need to be there, like this weird little micro tail looking thing, they go through apoptosis. So you remove those cells. You remove the webbing between what are going to become the fingers so that you end up with distinct fingers. And so that is apoptosis. You also see apoptosis in cells that are really old, and your body says, we don't need those cells anymore. They're damaged. They've picked up disease, and they picked up viruses. Let's just make sure that cell goes through apoptosis and we get rid of it so we can bring in new cells. We also, when we are going through division, we talk about differentiation, which is when you have a cell that could be something, so we need to make sure it's going to be what it needs to be, and that is differentiation. We'll get to it. So most of the cells in our body, almost all the cells in our body go through mitosis. And mitosis is where you take one cell, and then all of a sudden you have two cells that are identical at the very end. Okay, so you are making two cells from one exact copy of the, sorry, you have one cell, you make an exact copy of it, and you end up with two cells. So DNA is going to double once through DNA replication, and then the cell divides through cytokinesis. So cyto means cell, and kinesis means movement, whatever. Or you can think of this as cytoplasm moving to the other side. Whoops. Here we go. Boom, there we go. So mitosis is very important. Your DNA, in order for your DNA to replicate, it has to condense into chromosomes. So it goes from this spaghetti noodle looking thing into these big X looking things. We actually have 46 of these, and they once they look like X's, they've replicated. Before they replicate, they condense into just a single part, and then you can tell they've replicated because now there's two of each part. And then they line up in the middle here. And you have these microtubules, that cytoskeleton, that go and they reach out and they each grab one chromosome and then they rip them apart at what's called the centromere at the very center of that chromosome. Once they've ripped them apart and taken them to the other side of the cell, they can go through cytokinesis and you end up with two cells. And this is very important that these divide really carefully because you don't want to end up with like both of your X chromosomes on one side and both of your some other chromosome on this side. They need to be identical when they do this. So the DNA doubles and then the cell divides twice. Oh, sorry, I'm looking at meiosis now. Ha. So that was mitosis. Let's look at meiosis. So the difference between mitosis, which I like to think of, it happens in the toes, but it happens all over the body. But meiosis only happens in the ovaries and the testes. So these only happen in your gametes, or the eggs and sperm cells. Okay, so the DNA doubles twice, or sorry, the DNA doubles once, but the cell divides twice. So you end up with cells that have half as much DNA as the original cell, which makes sense. When we're doing mitosis and we end up with an exact copy, we're just copying skin cells, we're just copying blood cells, or whatever. You have an injured cell, you need to make a new cell, it better be an exact copy. On the other hand, with meiosis, you have, let's say you're a female and you're making eggs. You want to make sure that when your egg encounters a sperm, you end up with the right amount of DNA. So gametes, or eggs and sperm, have only one copy of every gene. So they have 23 chromosomes. They have one each of 23 chromosomes. Then when an egg comes in contact with a sperm, it suddenly has two copies of each and ends up with 4 chromosomes total. Sorry, 46 chromosomes total. All right. Again, the central tetrad is really important. That's where you have the maternal copy of something and the paternal copy of something come together, and then they split so that one side gets maternal, one side gets paternal. And the splitting is really important. If the cell doesn't line up properly, let's see, there we go. So here are cells that are stained through confocal microscopy, so a kind of laser microscopy, so that you can see the nucleic DNA here in red, and you can see the cytoskeleton in green. So we're going to watch it a few times, because let's see what's happening. These two cells are about to reproduce. They're about to divide. And so all the DNA lines up in the center part. If it doesn't line up, it won't split. So if you look over here at this one, the DNA is getting pushed to the center, pushed to the center. You see, this one never lines up. And so because it doesn't line up, the cell can't divide. Because if those chromosomes are not all centrally lined up, so the cell can basically check itself and say, I've got one copy of this, one copy of this. Okay, let's spread them out and do cytokinesis, no problem. If that doesn't happen, the cell usually will not divide. The times it does divide still are on the really small copies. So, okay, the problem here, let's talk a little bit, go back a little bit. So sperm cells do this every single day. They do meiosis, the progenitors of sperm cells that are in the testes are creating new sperm. As soon as you hit puberty, basically almost until you die. Every single day you're going through this over and over and over. But female cells basically stop right here. So the egg cells stop at what's called metaphase. And they say, okay, go. And this happens before you're born. So before you're born, all of your egg cells wait at this certain stage. And they just sit there. They're like, okay, we're going to wait until puberty. And then once every month, 1, maybe 2 cells will go through the rest of meiosis to create working egg cells. So they don't look exactly like this, but we're going to go for it for now. Now, if you have a cell that, let's say, two very big chromosomes don't separate properly, then this egg is not going to become a working egg cell because it can't say, yes, these are identical cells. I have the right number of DNA in each one. We're good. However, so imagine you're a woman and you have ovaries full of cells, egg cells that just look like this. They're ready to divide and become the final egg cell, and they're sitting on these really thin, very delicate microtubules just waiting to finish the cell division. And then you go through like 30 years of life with ovaries full of stuff like this, waiting to be used. It's very easy to see that over time these spindle fibers could break. And so when they break, and you have something like this over here where this one chromosome never wants to go to the center because its microtubule is broken, if it's a big enough chromosome, this cell will just not become an egg, will not become eggs. However, sometimes those chromosomes can be so small that the cell doesn't notice it. And this is where trisomy 21 comes from, or Down syndrome, where you have an extra, very, very, very small chromosome, but it's clearly enough to cause some major physiological differences. And so, yeah, we'll go more into that more when we get to reproduction. All right. But basically, the things to know here are for mitosis, almost every single cell in your body does this, and it does it for growth and development and replacement. Whereas in meiosis, this is the DNA splits twice, so you end up with cells that have half as much DNA as the parental cell did. This only happens in eggs and sperm. All right. So what is the cell normally doing? It's not just sitting there reproducing all the time. What it's usually doing is just going through its normal cell cycle. So it's doing stuff. It is, let's see, where are we? So at G1 or gap 1 is just normal cell life. I am a skin cell. I am protecting. I am a muscle cell. I am contracting, whatever. And then all of a sudden it says the cell is given a signal that says we should probably reproduce. We should probably make another cell. Let's start synthesizing DNA. And then it synthesizes the DNA, and then it spends some more time being a normal cell, having no problem. And then all of a sudden it gets a signal that says, oh, we've synthesized the DNA, let's split. And so it goes through mitosis, and suddenly you have two cells that are going through gap. Okay, that are just cell cycle doing its thing. Okay, there's some point where they stop getting the signal to go into DNA synthesis, and they either stop dividing, or they go through apoptosis and they go through programmed cell death, and they aren't used anymore. So we do have non-renewing cells, like there are some nerve cells, if not all nerve cells are still on the fence there, that will not renew. Basically, you have all the DNA, in theory, as far as we know right now, you have all the nerve cells you'll ever have, but they can make new connections all the time. On the other hand, the egg cells, you are born with all the egg cells you could possibly ever have, and that's it. Good luck. It's to kind of just whatever. It doesn't not work, so evolution went with it. And so then the stem cells that originally made those egg cells don't get replaced, whereas the stem cells that make sperm cells are constantly being replaced. All right. Mitosis only happens about 5% of the time. Most of the time, a cell is just being a cell, which makes sense. Cancer, on the other hand, instead of going through a phase where it says we shouldn't reproduce anymore because maybe we are growing in a layer, and if we reproduce more, there won't be any room. So cells sometimes get signals from each other to say, "Don't reproduce. I'm here. Don't get in my way." A cancer cell might not do that. So cancer is defined as unregulated cell growth. So in this case, the cell skips its checkpoints, and so whenever your DNA picks up a bunch of errors, usually the cell says, "This DNA is full of errors, so this cell is no longer functional. Kill it," and it gets rid of it. In the case of cancer, it doesn't do that. It either doesn't get that signal, or it has genes that actually cause this to happen faster, which is called an oncogene. So an oncogene is a cancer-causing gene. It's a mutation in a normal gene that then just causes cancer to happen faster. So if you have cancer that runs in your family, it could be that you have an oncogene. So that's why you were asked that question a lot when you go to the doctor. Sometimes you have an inactivation, so a genetic error that says This could be another thing that causes cancer to run in a family. So it's either you speed up something, you speed up cancer cells, or you slow down the inactivation of cancer cells. So if this were a car, then an oncogene would be like an accelerator, making it go faster and faster and faster. Whereas the inactivation of a suppressor gene is like having no brakes. Like you're trying to get that cell to stop reproducing and it just doesn't. So this could be something as simple as you have a cell right next to another. It's packed in between a bunch of other cells, and it shouldn't be reproducing because there's no place to put its baby cells. Its daughter cells. But it just does anyway, and so that would be a cancer. Let's see. Mutations that alter adhesion proteins allow cancer. All right. So then, there is a difference between a benign and a malignant tumor. So if you have a tumor growing somewhere, if it still has these what are called adhesion proteins that cause it to stay where it's supposed to be, like the lining of an organ or the lining of your capillaries or whatever, if those adhesion proteins are still there, the tumor will stay in place and it's considered benign. And that can then just be you can go in and take it out surgically, and so a lot of times you just don't have to worry about it anymore. However, if those adhesion proteins have a mutation and it should be staying in place and then all of a sudden there's nothing to keep it in place, you get a malignant tumor, and then they can spread and they can spread to other organs. And if you have like this one cell goes and launches itself into the bloodstream and then lodges itself in the lung, that would be, it could be a case of a cancer that has spread somewhere. So a malignant cancer is much more dangerous than a benign cancer usually, because it can go through the body. You never know if you've got everything. And so that's why a lot of times you go through chemotherapy for malignant cancer, because what chemotherapy does is it kills fast-growing cells. And so that includes hair cells, and so that's why people who go through chemotherapy lose their hair cells, because hair cells grow quickly, but you know what? So does cancer cells, and so do cancer cells. It's a lot better to kill those cancer cells and then just get your hair to grow back later. Alright, so if we have all the same DNA in all of our cells, and this is cancer cells or just normal cells in your body. So if you start from one cell, from an egg and from a sperm that come together to create one cell, and then you go from this single cell to this magnificent multicellular organism that is you, how does each cell know what to do? How does a skin cell know to become a skin cell and not a muscle cell or not a lung cell or whatever? This is called cell differentiation. So all of that DNA is packed into every single cell, but cell differentiation says we only care about a certain part of the DNA because we are a certain cell. And that happens through what are called cytokines. Okay, so the cell differentiation commits a cell to a certain fate. So you are going to be you are going to be a bone cell, you're going to be a cardiac cell, you're going to be a toenail cell, whatever it is, that cell differentiation tells the cell as it's being produced as a stem cell what it's going to be. So there's different levels of commitment. If you're a totipotent stem cell, this is a cell that could become anything. And people have been really interested in this research-wise, because the idea is if you have a cell that has the potential to be anything, if you give it the right cytokine, you can turn it into anything. So if you have just a big old bowl full of totipotent stem cells, and then all of a sudden you need a liver, you could give it a liver-determining cytokine, and then those totipotent stem cells would become a liver cell. Most of the time, we don't have a lot of these because it's really easy for this signal to get messed up. So you can imagine these things. You don't have a lot of these because it's very easy to tell these to do something that they're not supposed to. So this happens usually in fetal cells. In pluripotent stem cells, these are committed to a certain kind of fate. So a pluripotent stem cell could be like all of the blood cells. So you have a pluripotent stem cell, and you say, you know what? This is blood. This is the lymph system. So you have a pluripotent stem cell that says any cell that gets made out of this has to be, so first it was totipotent, and then it was given a signal that says you're going to be an immune cell. You just don't know what kind yet. And so then you might get a signal that says, well, you're going to be a lymphoid stem cell, or you might be a myeloid stem cell. And then you either go and you become a blood cell, Okay, so this actually isn't lymph. This is something different. Or you're going to become a lymph cell and be used in the blood. The immune system. So this cell cannot become something else. It could be given all of the, especially if you're here, you could be given all of the signals to become a bone cell, but that part of the DNA has been turned off and hidden away. Okay, so it's only going to respond to signals that turn it into the something in the lymphoid family. So a thymus, so a lymphocyte, a T lymphocyte, etc., etc., etc. So the thymus is an organ. So if this goes to the thymus, the cell, there are signals there waiting to turn it into a T cell. If it then goes to a different part of the lymph system, there are signals waiting to make it become a plasma cell or an NK lymphocyte, which is a natural killer cell, which sounds like a crazy rapper. Crazy good. Maybe crazy bad. We don't know. It's not a rapper, but it sounds like it should be. On the other hand, our marrow, our bone marrow, contains myeloid stem cells, and depending on what signal they're given, they'll either become a red blood cell, which is called a erythrocyte. They could become white blood cells, which is it's like a macrophage. They could become a clotting element. There's so many things that it could become. And if you think about it, the blood cells and the lymph cells are the ones that we use, and they're moving through the body all the time, and they're encountering a bunch of stuff. So we need a lot of it. We need it to be able to respond quickly to what we need. So it makes sense that these cells are waiting for signals to see what they should become. They're like emergency response cells. Whereas like a bone cell that's in your bone better not change it into anything because your bone is there for a reason. So that's a lot of information, but I want to show just kind of, we're going to go through little parts of this video. And this is put out, I think it's by Harvard. I'll put a link to it. And it's basically showing, so first we have a bunch of blood cells traveling through a blood vessel. And this is a white blood cell going around looking for diseases. If we get a little bit closer, let's go on to that. If we go into like the cell membrane, so if we're approaching a cell membrane, so that's what this bottom part is. We have these things called lipid rafts that can float on top of the plasma membrane. And these could be signal proteins that say, hey, you know what cell this is? This is this cell. It's awesome. So you can see the plasma membrane here. Here's the, I can't even think of the word. The hydrophobic end is on the outside, encountering either the extracellular matrix here or the intracellular matrix on this side. But you can see stuff can pass through it. It can also float on top of it. So the cell membrane is very fluid. It can move a lot. Let's see what else. So now we've moved into the cell. Let's kind of let that transition happen. On the outside of the cell, you can have receptors that then say, hey, make this thing happen. And then if you move through the plasma membrane, you can see what's happening. So we've moved through the membrane. Now we're on the interside of the cell. So we used to think that the cells were just bags of fluid. We now know there's actually a lot of stuff happening. You have these transmembrane proteins that let stuff happen. You can have proteins that are anchored into the plasma layer. Let's keep going. But you can see these strands we see are those microtubules that are, don't worry about this word, but they're creating structure inside the cell because the cell itself doesn't have structure. It's a big globby bag. But the cytoskeleton can create structure. So if we keep going deeper into the cell, you see more and more tracks. And these are the microtubule tracks of the cytoskeleton. So not only does it give the cell structure, but it lets, so this is they self-assemble to make these certain pathways. And so there's some more cytoskeleton being produced. It's very exciting, very pretty. Again, this is computer simulation, by the way. We can't see this yet. And then we have these proteins that come in and say, no, I don't want you to be there. And it severs that cytoskeleton. This could allow the cell to move. It could allow the cell to get ready for cell reproduction, cell division. It could make way for different tubules, like these microtubules. So you can tell there's a bit more structure to these. They're bigger. They're less for like just holding the cell up and more for allowing stuff to walk on it and move around the cell. So here's a microtubule that's disassembling for whatever reason. We're going to keep going. Oh yeah, this is my very favorite thing ever. So here's a microtubule. Here's a vesicle, which is full of proteins or whatever. And here you have my most favorite molecule, Big Pimpin', the actin motor protein just moving. And so what it's actually doing when it's doing this, it's not actually walking because it's not a live organism. But what happens is you have areas where all of a sudden it bonds, and so it looks like so it gets stuck, and then it unbonds with the next foot can move forward. And as it moves forward, it pulls that vesicle with it. So let's look again. My favorite, the motor protein, just zooming along with its big old vesicle. You see one back here too, moving that vesicle along so that it gets to where it needs to go a lot faster. These weird bug-looking things are the mitochondria. And when you look at it this way, it really does look like a different organism. But it's making a bunch of energy for us as well as our steroids, so they're very important. These are the microtubule centers called the centrosomes. They shoot out a bunch of microtubules to make sure the cell is staying in the right shape. And also, once the cell goes through division, it'll move the DNA to separate sides of the cell. Let's see. So here's the nucleus and the pores coming out of the nucleus. So these little things coming out are the messenger RNA. And here's the endoplasmic reticulum, the rough endoplasmic reticulum you can see because it's got the ribosomes on it. Here's a ribosome that just assembled. Here we go. It's reading that mRNA and it is popping out a protein chain as it does. And that protein chain just left. It's going to go. So there's another protein chain. Oh, look, it's assembling with another protein chain that it's supposed to bind to. And it goes to, there was a mitochondria, but it goes to the ER so it can be processed more. So it's going to go and it's going to die. Don't worry about this. But it's basically, this is where the ribosome says, hey, endoplasmic reticulum, here's some stuff for you to start figuring out. So so it's not pooping. It looks like it's pooping. It's creating a protein chain, a peptide chain inside of it. So then the endoplasmic reticulum can say, hey, what do I need to do with this thing? And so it starts gathering them together into these vesicles, which it then plops off. And then that motor protein, my very favorite thing in the entire universe, can grab that vesicle full of proteins, lots of different proteins, and take it to the Golgi apparatus so the Golgi can start sorting it. Here's our Golgi. It's more blobby looking because it creates more vesicles that are more specific. So there's that vesicle that came in originally. It gets just kind of absorbed into the Golgi, and then the Golgi sorts all those proteins and then sends them again off on another tract to go and see, there's our motor protein full. Here's a vesicle full of very specific proteins. The motor protein pulls it along that cytoskeletal tract to wherever it needs to go. And then it plops it off, and so it absorbs, so that vesicle just kind of absorbs into the plasma membrane and throws whatever was in it out into the extracellular matrix. It gets so much more complicated, but we're not going to go into it. Just make sure that you know the cell molecules, the cell organelles we went into today, and also go over the differences between meiosis and mitosis, and then what a cytokine is, and the difference between the benign and the malignant tumors, and you'll be good. Bye!